All right, so I'm going to go through this a little bit in chronological order of what we did in the class, but not entirely. Some of it's going to be mixed together because some of the questions I have on the exam are like combination questions of two ideas coming together, something along those lines. And so I want to show you a couple of those things about how to connect the ideas.
Okay.
So let's, we can look at the PDF first. So, excuse me. Some of the themes we looked at, structure function, which we'll talk a little bit about later. So I'm just on slide 2, the domains of life. So you're looking at bacteria, archaea, eukarya, that kind of thing. It's pretty straightforward. And then now we've done at this point, you know, you understand gene expression, which was something we saw in the beginning. You understand cell division, which was something they had in the themes. So those are all kinds of things that you kind of need to know, but I'm not having a lot of questions about themes.
Okay.
I do have more questions about, if you go to page 3, about like scientific experimental design, since we did focus on that quite a bit at the beginning. So it'll be things about hypotheses and what's that versus a theory, controlled experiments, negative control, positive control, independent variable, dependent variable, reading some graphs and interpreting the data there, that kind of thing. So, you know, you're just looking at the axes, you're looking at what it's showing.
Okay.
You know, just go through those. Because sometimes there's a lot of information on there, but if you just go through it stepwise, it should be okay. And you've practiced making graphs and things in your labs as well, so that should help you. Okay, so one thing to remember is that we support or reject or not support hypotheses. So a hypothesis is your proposed answer to a question. You know, always in science, you have some sort of question out there that you're trying to figure out what's going on, and so you want to propose an answer to that. That's the hypothesis. If you get results that do not support your hypothesis, we wouldn't say you disproved it. If you get answers that you do support your hypothesis, we wouldn't say you prove it. And remember, that's because there's always potential confounding factors that you didn't realize were there. So we'd say you support, or you can reject or not support your hypothesis. So that's one thing. So usually if I have an answer on there that has like prove in it, that's probably not the correct answer. Because, you know, like I said, you never know what else is going on. I mean, you try to design your experiments the best you can to make sure there are no other things going on. You know, you're not doing it willy-nilly. But, you know, we're not so egotistical that we think all of our answers are always perfect. So if you go to page 4,
okay.
Just a brief reminder of what can and cannot be addressed by science. So exam 1, I had some examples of, here's some questions people are asking, which are scientific and which are not. Remember, it does not have to be experimental. It can be investigatory, like you're observing something in nature, or you are looking at the fossil record or the genetics of something, you know, so it can be observational, but you still always have your hypothesis, you have some question, and it has to be based in things you can observe and measure in nature, right? So supernatural, which includes religious explanations, which are based in faith. I mean, the whole thing with that is that it's not evidence-based per se, right? That's like the definition. So that would not be a scientific explanation for something. So that's pretty straightforward. Let's see. Okay, if we go to page 5, remember, a controlled experiment means one that has one or more groups that I'd say you have either known outcomes and or they don't get the variable treatment or the variable factor, I guess. So that's what controlled is. It does not mean everything's constant. or that you're controlling all the stuff in the experiment. It just means that you have groups there that you're able to compare to see, like, when I change this factor, what happens? Well, you can't just change that factor and have nothing to compare it to. You have to have a group that doesn't have the factor changed, or a group that has a factor changed in a way that you know the outcome. So the classic examples, right, with drug trials are people that take the sugar pill placebo versus the drug, or take the regular regimen versus the regular regimen plus the drug. Like, sometimes you can't give people placebo for conditions because it's unethical, right? So in that case, you give them like the known treatment instead of this different treatment and see, does it improve the condition, right? So a positive control. Oh, and the reason for controls is to get rid of these unwanted variables. That's what I'm calling them on here. Ooh, that did not work. Let's see. Unwanted variables, or you can remember, we call those potential confounding factors. Ooh, it's writing really slow. Okay, potential confounding factors, those unwanted variables, that's why you're matching the groups. So one of the things that helps you verify the PFCs are not there, that you don't have any confounding factors, are, oh, there we go. So positive controls, which means that the outcome is known to be a certain way, like when we use glucose in, we use glucose to test the test strips worked. So when you have those test strips, you know you need to just use plain old glucose to make sure the test strips work. That's a positive control. We also use lactase plus milk, because you know that milk should digest lactase should digest the lactose in milk, so that was a positive control because you know the outcome should be positive. And if it's not, then you can't trust your results. Then you have negative controls where you know the outcome should be negative. For example, just testing sucrose by itself with no enzyme. Like, if you have no enzyme, there should be no reaction, thus you should have a negative result. And the same with milk without any enzyme, because you just have straight up milk, you shouldn't have any glucose in that. So those are negative controls. The group that has no drug treatment in the drug trial, that would be a negative control. They're giving you like a baseline. So that's, and as I mentioned, control does not necessarily mean everything's kept constant. It just means, like, people can go about their lives. You can't just keep people in a little, like, sealed chamber, or, you know, the birds outside you're studying, something like that, where humidity is constant and all those things. But you want to make sure you look at birds in the same area at the same time of year, let's say, unless those are the things you're studying. Or you want to make sure the people that you're using are all sort of like the same age. Sometimes it matters if they're males or females. Like, you might want to match those groups. It just depends on what you're trying to look at, but you do consider that. Okay. All right, just to practice independent variable, dependent variable. So let's look at this. The question is, does heating a cup of water allow it to dissolve more sugar? So the independent, right, and the dependent. Dependent is your data. Independent is what you're manipulating, or the difference between the groups. So what's the data you're looking at here? That's your outcome data you think you're going to measure.
The amount of sugar that dissolves.
Okay. And then what do you think is mattering? Like, what factor do you think matters?
The temperature of the water.
Sure. That's good. So temperature and then amount of sugar dissolved. Does an electric motor turn faster if you increase the voltage? So what are you going to measure there?
How fast the motor is turning, and your independent variable is going to be the increasing of the voltage.
Yes, and you could even just say the voltage. The voltage itself is independent. Yep, and then how fast it turns is dependent. Absolutely. And here's an example of a drug trial. So we have whether a new drug increases. I guess this is in rats. Running performance memory-based tasks in a memory-based task in older rats, just like aging humans, rats show a decreased ability for learning new things due to poor memories. He teaches two groups, or Dr. Smith teaches two groups of older rats to find a piece of rat chow in the maze. One group is given the drugs while they're learning, the other group is not. 1 week after Having learned the maze, he retests all the rats and records how long it takes them to find the rat chow. So I think this one's the easiest thing is to say, like, first, what's the data?
That how long it's going to take them to find the rat chow.
Right, and which variable is that then?
The whether they have the drug or not?
Yes, and so which one's dependent, which one's independent?
The independent variable is going to be whether they have the drug or not, and the dependent variable is going to be how long it takes them to find the rat chow.
All right, good. So the drug is the independent, time to find the rat chow is dependent, which is reflecting memory tasks. You know, it's how long it takes is reflecting their memory ability, but that's the actual measurement. Good. Okay. So that's, oh, and then I'm going to be on page 8. So hypothesis is that answer, the tentative or temporary answer to the question. So you have a question, and that's your temporary answer. So it's narrow, right? Like lactase is linkage, a linkage specific enzyme. That would be your specific kind of question. That's your hypothesis. Then we go to page 9. Then that is about theories. So theory, right, is not a hypothesis. It's a larger scale. It's like a big explanation that's supported by lots of facts that explains a large phenomenon. Theory of evolution explains how organisms change over time, the factors that are involved in that, why we see things the way they are today and in the past, and how those kinds of changes happen. Atomic theory says that all matter is made of atoms, and those atoms have certain kinds of characteristics. Cell theory says all living organisms are made of cells, and cells have certain characteristics. So those are all different types of theories. And you can see how much bigger those are than just a specific answer to a question. So you'd want to know the difference between those. The fork is just showing you all the parts that are doing the DNA replication. So first of all, remember DNA replication's job. It's sometimes kind of confusing because it's like fairly similar to transcription. But transcription is where you're reading DNA to make RNA, and it's just like a temporary reading process. You make a little RNA that's released, and then your DNA closes back up. DNA replication, on the other hand, is where you're making new copies of the DNA that are going to stick around and then be put into the new cell. So I'll draw some stuff too. So the idea here is that you have one double helix, and you're going to, in S phase of the cell cycle, copy using that one template, and you're getting that 2 double helices. It's a little bit sad, double helix, but you get the idea. And the reason I draw it in different colors here is because one strand is the original strand, the template strand, from the parent DNA. That's the first one. And this is not parent as in like, mom, dad. This is parent as in like the first original DNA molecule. And then each of these has one old and one new. And the new one comes from the replication. So the whole beauty of DNA replication is based off of DNA structure. So I don't think I have much, if anything, about the people that did the different discoveries. I mean, Watson, Crick, Franklin did the DNA double helix. Prior to that, we have Griffith doing, he's the one that first showed that there's a factor, they didn't know what it was. There's a factor that can be transferred from dead bacteria to living bacteria that changes the living bacteria's characteristics. So that showed that there is discrete material that gives characteristics to organisms. It's not magic. It's not just like, it's an actual molecule entity that can be transferred that gives characteristics. And then we have Hershey and Chase showing that what gets into the cell using the viruses and the E. coli, the bacteria. What the viruses put into the cell is nucleic acid, DNA specifically, not their protein, which suggests that, you know, DNA is the genetic material, not protein. And then we also have Avery doing some stuff where they cut up, they do Griffith's experiment, but they cut up the DNA and then it doesn't work. And they cut up the protein and it does work, which suggests that the DNA is necessary, because when you cut it up, now the transformation doesn't work. So those are some of the main ideas from the people, but like I don't have too much on that. Okay, so you have these double helices, one old, one new, and this is all based off those base pairing rules, right? So the straight letters go together, the A and the T, and then the C and the G, and it doesn't matter which order, and they have hydrogen bonds between them. There's 2 versus 3. And that's what holds the helix together. That's happening between those 2 double helices. So each of those helices is made of long strands of C's and G's. And the bonds along the strand are covalent bonds. The bonds between the strands are the hydrogen bonds. That happens because of the nitrogenous bases. So each nucleotide is made of, DNA nucleotide is made of a deoxyribose, which is a pentagon shape, and it has an OH and an H. It has a nitrogenous base over here, so it's some rings with nitrogens in them, so that's your ATCG part. And then it has a phosphate group. It starts with 3 phosphate groups, but in the end, once it's in the strand, it has just 1, which is why DNA is negative, because phosphate has negative charges. And the phosphates are going to attach to the oxygen that they build on this oxygen here in the next nucleotide. So then I have 2 nucleotides attached together. Let's just say we have the A and then we have the C nucleotide like we did there. There's that phosphodiester bond, that covalent bond between the 2. And then the hydrogen bond would happen with the next strand. Remember, the strands have polarity. So why is there polarity? And this is important because it's why they're built in different directions when you're replicating. So polarity just means that there's a side that has a phosphate group on it and a side that has the hydroxyl group on it. So the way that we talk about polarity is based off of the carbons. So these guys have 5 carbons in the molecule, 4 of them on the edges of the hexagon or the pentagon, and then 1 sticking up. I made them in these dots. So just based off of chemistry, there's ways you label carbons. So it's a 1' carbon is over here, then the 2', and those don't matter so much for our naming, but 3' does. And this is the 4' and this is the 5'. So that's why this phosphate end is called the 5' end, and then the oxygen or the hydroxyl group is the 3' end. And this matters because both DNA and RNA, for that matter, only can be built on the 3' end. And that's why they are built directionally.
They can only be built on the 3' end, but they go 5' to 3'?
Right. So I for a long time I also was like, wait, the way I remember it is it only builds on the 3' end, which means that which direction does it build? Well, if the 3' end is what's getting added onto, think about which direction the whole thing has to be moving. If it's the 5', 3', it adds onto the 3', and then it adds onto the next 3' and adds onto the next 3'. That means it's moving from the 5' to the 3'. So yes, it's being built in the 5' to 3' direction, but that's because it only adds onto 3'. So every time it would add on to, in my case, the left side. But you can have orientations like this. And in that case, it would always add on to this 3' and we'd be building in that, the 5' to 3' direction, but it would be to the, quote, left as far as my drawing goes here. Okay, that's why. So this is true of both all polymerases. So DNA polymerase, that's what you build new DNA with, can only add on to 3' sides. RNA polymerase, which is transcription, so that's what you build RNA with, that can only add on to 3' sides. So let's look at this bottom picture on this slide. I'll draw on the slide and then I'll erase it because it won't stay on the slide. But and if we look at the polarity here, now how are you going to do this? Well, you do this by either starting at the arrow or starting at the not arrow. So I like to start at the arrow because the arrow is the direction it's building. And now we know what must be at that side if that's the direction that's getting added onto. Like, for example, this one here. What does that tip have to be of the arrow if that's where the building, I call it the action, where the action is happening? Does that happen at the 5 or the 3?
The 3 end.
The 3 end. So that tells us the 3 end, which actually means any arrowhead is a 3 end here. And even if I didn't have the arrowheads, how are you going to tell? Well, we've got 2 different colors. One color is the red and the other is the light blue. So what's the red? Do you know what the red is? What's that little piece they make at the beginning of the replication? It's okay if you don't know. Just
wondering. I don't know.
All right, let's go look up at this little picture up in the other corner. Okay, so you'll notice here it's the opposite colors, but we have red and we have blue, and the little piece is the blue piece. So that blue piece is just like when you're painting your walls, how you don't want to just straight paint your walls. You want to put that primer on first. Or a lot of people use like primer under their makeup, right? It's the first layer. And that's what these blue pieces are called. These guys are primers. They're to prime something means to get it ready. So those are the primer, which is RNA actually. It eventually gets taken out, so you don't have any RNA in your DNA. But it's probably RNA because that was the first genetic material, we believe. And so this is a leftover version of that. And why is that happening? Well, DNA polymerase can only add on to a 3' end. RNA polymerase can add on to nothing. Its shape allows it to do that. But DNA polymerase must have a guide end to add on to. So we make the primer to prep the DNA so that DNA polymerase can jump on and start building that strand. And so the primer is always a small part versus the DNA is going to be the big long part. So if we go back down here to our picture in the bottom right, which one is the primer part, the light blue or the red?
The red.
The red at the beginning of each little DNA segment, whether it's a long segment or little short segments. And the primer is the first part, so that end where the action is no longer happening, no action, is the opposite of where it's building. Right, so where it's building is the 3', which means the opposite end must be, so like this one here, which end is that red one there, or all the primers? 5' or 3'?
The 5'.
5', the opposite of the end that's getting built on. So all the primers are 5' sides. Now that means we can also figure out the polarity of the parent DNA. And you'll also notice too that we have anti-parallel strands here. 5s are opposites 3s. So that's just because that's how DNA strands can fit together. So it's going to end up being a phosphate on one end and hydroxyl. And so actually, what does the 5' mean? That means there's a phosphate on that end versus the 3', which means there's an OH on that end. So, and that's really why the reaction happens there, because the OH is needed for dehydration synthesis. Remember how we pull out of water and stick 2 bonds? So you need the OH there because the phosphate group doesn't have that. It only has oxygens on the phosphate. So that's truly why you can only build on the 3' is because it's a dehydration synthesis reaction happening. So if we go to this guy over here on the top left, the parent DNA, what I like to do is I look at who is building towards it, or who's going to end up over there. So it's going to end up building out this way, right? Let me make that light blue. This is working better. I'm glad I closed those programs. I don't know what was happening. You see how it's eventually going to build out this way?
Mm-hmm.
So what is this end over here that I just drew the arrow at?
A 3'?
It's a 3'. So what does that mean the parent DNA is at that side? They have to be anti-parallel.
The parent DNA?
Yes, the one that this dot one here.
Are you asking me whether it's like a phosphate or
so you can say whether it's a phosphate or hydroxyl,', or whether it's the 5', 3'. Both mean. So
a 3'.
Okay, so why would be the 3'? What's your logic?
Because it's building on that side.
Oh, the 3', yes, right here is the 3' because it's building.
Mm-hmm.
Now what about
the other one?
Yeah, what about the parent DNA in the dark blue?
The 5'.
Because it has to be anti-parallel. So if the 3' end is going to end up there eventually, once it builds out, that's the 5'. Then you know everything else, because it has to be, the parent DNA was originally anti-parallel. So if that's a 5' on top, then you know what has to be the bottom strand of that dark blue DNA. If you have a 5' on the top side.
The 3'.
It has to be the 3', and I'm going to do that so we have more. So this has to be the 3'. And then those strands have polarity. So if we trace it all the way back, and we go back to the other side, over here, if the 3' is on this side, what must be on the other side then?
The 5'.
Must be the 5. And then opposite that must be the 3.
The 3.
Exactly. And does that match with what we have on the left side? It does, or it's opposite, 3 and 5, anti-parallel. And then also we can check, because does our 3 here match up with what will be a 5? Yes. And you're like, wait a second, that's not going to grow backwards. You're right. That's why it has to build like this in little baby chunks.
Mm-hmm.
Because it can only build in the opposite direction as the fork. And you think, well, why is it going continuously on the top? Well, this strand is building on the 3'. So it just keeps going. So that long strand there is the leading strand. So leading strand is this top one and this bottom one. Those are 2 leading strands because they're built continuously. And the lagging strands are all these little chunks, the Okazaki fragments, named after the scientists who discovered those. Those are lagging strands. And the reason they are on both of them, leading and lagging on both, is because you have 2 forks, and each fork moves in the opposite direction. So you have one fork here, and that fork is opening up to the left, and then you have the other fork here, and that fork's opening up to the right, which means that they're building in those directions.
Mm-hmm.
Now, why does that happen? Well, like, why does it have to build?
Sorry.
That's okay. No, I was not cursing at you. I was cursing at how I want to say this. So why does it have to build like that? Well, if we actually go look at this top picture, it's not a complete picture. A key feature here is that the helicase, which is what's opening the helix. So how do you know what helicase is? The helicase sits at the helix and it unwinds it. So which one's right at the fork? Which one's right at the fork there? What letter?
I can't see the letters. They're too small.
Okay. Do you want to zoom in? You should be able to either use your mouse scroll or your...
Okay. A.
A. Okay, so that's helicase. We know ACE means enzyme, and helix is this, twisty. So helicase is the untwisting enzyme. Now, the thing is that they didn't draw on here is actually your DNA polymerases are pretty much attached. They're attached with another molecule to the helicase, which means they can't really get too far away from the helicase because they are reloaded back on. So it's like they grow. You can see that's not a problem for this DNA polymerase down on the bottom because it's growing. The 3' is on that end. It's just growing, growing, growing. The helix will open. It'll grow, grow, grow. The problem is this helicase or this DNA polymerase on the top, because as we see, it's going to the right. So if it's attached to the helicase is opening. It's going the opposite. So it only builds a little bit until it's reloaded back at the fork, and then it builds a little bit more until it hits the one that it used to, like the old one. I mean, the one it just built. Then it gets loaded back and goes until it hits the one it just built and goes in that. It's sort of like if you've backstitched ever. I don't know if you've ever sewn, but you go like up, down, and then you come up behind the first stitch and you go down in the hole you made. You come up behind the second stitch and go down in the hole you made. It's like this idea of, so that's the leading and lagging strand and why that happens. Okay, let's look at who's making the little blue parts here. So after helicase opens it, we have kind of like two things happening. We have these little pink things. So the pink ones, which is H. Yes, H. So the little pink balls, they're actually keeping the DNA from going back together, because the DNA, the hydrogen bonds, they would just stick back together. You got to have something there to prevent them from sticking back together and resealing the helix. So these are just single strand binding proteins because they bind the single strand. And then you can see the single strand binding proteins come off, and you have this guy here. This is B. So B, it's making the little, and really this should be this is not the greatest picture. This should be sitting up at the tip. So it's actually at the place of where the action's happening. It's building this little, I never saw that before, that it was on the back side. But it's building the little first fragment. So remember what we called those first fragments? To get the DNA ready?
No. But I know that's primase, I think.
Uh-huh. And so what would primase be building then?
I don't know.
Which part of the name means enzyme?
Ace.
Okay, and what do you have left?
Primers.
There you go. Primase builds the primers. So those little blue things here, those are all the primers, which means to get something ready. So those are the primers, and indeed, that's primase, which builds the primers. Mm-hmm. So primase acted a bunch of times to build these new primers. So that DNA polymerase, so we have two of those. Those are the green ones. So I, and then the one at the top. DNA polymerase is building the DNA, which is in red. And this is DNA Pol 3. Usually I try to give you a clue as to which one is working first or second. This is in bacteria as well. So when you take genetics, you'll learn about this in eukaryotes, and there's like 5 DNA polymerases, and they have Greek names. But we're just learning about the bacteria. They have 3. We don't even talk about number 2. But DNA Pol 3 is the big builder. So that's building your DNA. Then, and so what do we come to here? Let's see, which one's the leading strand and which one's the lagging? So E or F, which one's leading and lagging?
F is leading and E is lagging.
Yes, good, because it's the short, or it's the continuous versus the discontinuous. Okay, and then we have these 2 little guys here, and this is just one of the 2 forks, right? We're just looking at the left fork. This would actually be continuous here until it got to the other fork. All right, so we have to see what's going on. Well, one clue here is that our letter C, the yellow one, is sitting at the area between the primer and the DNA. So that's where it's acting, is the primer and the DNA. Versus this blue one, letter D, is sitting right at this place where there's the gap. So both of these are enzymes, so they're both ACEs. So two things have to happen at the end. One is that we need to get rid of the primers because we don't want RNA in our DNA, right? That'd be bad. So we got to get rid of those primers and instead add DNA. So we got to chop out the RNA and replace it with DNA. And then the last thing is that the fragments, we need to seal them back together. We need to make that last bond called a nick. A nick is just a single break in the DNA, so we on one strand. So we need to seal those nicks up, bind them. So which of these, C or D, looks like it's taking out primers and putting in DNA? Who's acting at a spot with primers and DNA?
That's C. And then that is DNA polymerase 1.
Exactly. And you can remember it's a polymerase because it's adding DNA. So anytime we're building with DNA in a cell, it's a DNA polymerase. In viruses, reverse transcriptase can actually build DNA as well, but that's a virus thing. But in cells, DNA polymerase is what builds DNA. And Pol1, exactly. And then this last one, which is an enzyme, so we know it's an ACE.
It's ligase.
It's ligase, and you can remember that because ligature means to tie. Like if you ever watch the CSI shows, they have like ligature marks if someone's gotten strangled with like, like shoelaces or something. That's what that means, is that it's from something tying around. So that's what to ligate something is to tie it together, which is where ligase comes from, the tying enzyme. And then the last one we have here is letter G, which is sitting in front of the fork. And you can see that they've drawn this little curlicue. That's representing that the DNA is always twisted in a helix, and it's always twisted on top of itself, all those levels of wrapping. And when the helix is opening, it actually wraps it more. Give me 1 second, I can show you. Is this going to work? Is this too small? It's too small. Hold on. Okay, it's like this idea. So you have like a double helix here, and if you're opening the helix, it starts getting twisted on top of itself. Can make it do this. You see how this is like twisting on top of itself? The hairband, because it's a double helix, but when you push a helix, it just starts to do what's supercoiling. And so you can see that it's now really twisty on top of the twist. So it's twists on top of twists, and eventually it'll be too twisted, and then it'll break, and then your DNA will be in trouble. Because like the whole thing's broken. So that's why we have to do, so what happens is, as it's twisting, there's an enzyme that cuts it in front of the fork so it can unravel a little bit, and then it seals it back up. And then so as it untwists, it's going to cut it, allow it to relieve some of that supercoiling. It's like coiling on top of coiling. And so it's an enzyme as well because it's doing a reaction, so it's an ACE. And this one comes from the word topology. I don't know if you remember in geometry or something like that, one of your math classes, you looked at like, or in geography, you looked at the topology of the earth. It means like the shape, like mountains and valleys and flat plains. That's topology. So I sort of remind myself that it's like the shape of the DNA, the folding, coiling. So that's, I believe, where the topo comes from. And then where does the iso come from? Or where does the isomer come from? Isomers means the same.
Say that.
Say what?
How do you say that enzyme?
Topoisomerase.
Topoisomerase.
Yep. And the isomer comes from the fact that you're not actually changing the DNA. It's iso meaning same, like isosceles triangle, same. So the topoisomerase does the same topology. It's or it changes the coiling, but it's leaving it not changed in the end. Topoisomerase. Topoisomerase. Okay. So those are the parts that are working in the DNA replication fork, basically. That's why it builds 5' 3', old strand, new strand, double strands, covalent versus hydrogen bonds, polarity thing, happens in S phase. And it's just working by the DNA polymerase just matches up A's and T's and C's and G's. And this is when the mistakes happen that lead to mutations. So usually the cell, if it makes a mistake, there is proofreading. So the DNA polymerase can double check the shape is correct. And if it's not, machinery will come in and cut it out, and then the polymerase will try again. So almost always, we have billions of base pairs, and almost always all the mistakes are recognized and fixed. But on occasion, there's a few that get through, or there's some sort of disaster, and it's like the cell does emergency mode, and it's just like putting in nucleotides because the cell's starting to divide, and the DNA is now replicated, which is like really bad. So it's like, okay, emergency, we'd rather put in some nucleotides than nothing. So then you can get mutations like that. And so that's what leads to mutations, which are not necessarily bad or good. They end up in the next cell. And when they're in the next cell, the cell doesn't know. The cell has no idea that anything's different than it used to be. So it just reads them like it would normally read them to make RNA, and then the RNA is read to make the protein. Or it uses them for regulation. And so whatever is different about them then just becomes in that cell forever. And if it's in a somatic cell, a body cell, then that would affect that organism. If it's in a cell that is a reproductive cell, then that can affect the next generation and how the characteristics are for that. So that's why it matters whether it's in a reproductive cell, germline cell. That affects the next generation. That's what is for evolution, new phenotypes, that kind of thing, versus the body cells, which can be like lead to nothing, or they could lead to something like tumors, cancerous tumors, things along those lines. Or if like someone, some people have two different colored eyes.
Yes.
Occasionally that's because they got like punched in the face and there's damage to their eye and one of the eyes turned blue. But if someone's born with two different color eyes, usually what happens is that after their cell population goes to the two sides of the embryo's head, one of those, and this is only a few cells at this point, one of those cells gets a mutation that, because usually one of the eyes is blue or green, and so that turns off the gene for the brown pigment, and so then you end up getting one eye that's brown colored and the other eye that's green or blue colored because it got a mutation really early on, and then that led to a different color eye over on the other side. So that would be an example of a consequence of a mutation and some sort of cell division. Okay, so make sure you know PCR, polymerase chain reaction, and how that works, the different stages of it. It's something from 13.4. It's basically like synthetic DNA replication. So instead of helicase, you use heat to undo the double helix. Then you've made primers, like you make your own primers instead of the cell making them, so you don't need primase, but make your own primers. They bind to the complementary sequence during annealing, and then basically you put DNA polymerase in there, and the DNA polymerase jumps on the end of the primers and builds until you heat it up again, and then the new helices come apart, and then the primers jump on, and then the DNA polymerase builds, and then the new then it keeps on going, and it just replicates until you have like 1 billion copies of DNA. So that's the process of polymerase chain reaction, PCR. It's really important. We use it all the time in the lab. And it's really important because like you only need a tiny bit of DNA in order to make a lot of copies of DNA, which can be used for a variety of things. This is why all the forensics can be done you know, you only need a tiny piece of DNA in order to like identify somebody or identify a suspect. This is also why a lot of lab experiments can be done. It's why we can build our own DNA. I mean, it's all kinds of things. Oh, and one thing that's special is that I mentioned it has to heat it to do the denaturing, which means the DNA helix coming apart. Well, normally if you heat it up a lot, your proteins are all going to denature too, right? Because they'll unravel. So how do you prevent that from happening? Well, they got real smart and they picked a polymerase to use that's from a bacteria that lives in the hot springs. So hot springs bacteria have modified polymerases that do well in heat, and they don't fall apart, they don't unravel. So that's the polymerase you use. And we have a couple different types of organisms, bacteria and archaea, that we've gotten those polymerases from, and that's the ones you can use to do the DNA, the PCR. And that's like the special part. That was the key. Because they used to do it, and they'd have to like every cycle put new DNA polymerase in, and it was really inefficient. Now you just put it in a machine. It runs for an hour or something. And this is also like you heard RT-PCR, qRT-PCR, all the COVID testing stuff. It's all based off the idea of PCR, but we're using RNA instead of DNA to do the copying of it. But it's the same kind of idea of the process. And you saw, I mean, how fast that can be done. And that's because we have this ability of the PCR.
That's cool.
Yeah. You need to understand banding patterns on a gel. So basically we can take DNA, put it into, like, it's sort of like Jell-O, and has little divots in it. And, but it's a little harder than Jell-O. So it's made of a matrix, like, sort of like, carbohydrate molecules that are all crossed and tangled together. So the way the tangles work is that it prevents big things from running very fast, but the little things can get through the holes. You could just imagine, like, if you had tangles, little stuff would wiggle through, whereas big stuff kind of like gets stuck. So that's how you can separate the strands. Because when you're doing PCR, you're only making a copy of a little bit of the DNA. You're not doing DNA replication entirely. You're just doing a segment. And so sometimes after PCR, you need to like separate different size fragments out because you're only looking for one of them, or you want to do like DNA profiling where you're looking at different size bands. And so in order to separate them out by size, you run it on what we call a gel. So the small fragments are going to move far in the gel. The big fragments don't move very far. And then you can look at the patterns of those. And so you can see here the crime scene and suspect 2 have the same pattern, which means they have the same size fragments, which means that those guys are matching, which would mean that if you do enough, like you do enough PCR products, we look at 13 sites. So you do PCR on 13 different parts of the DNA. And each part has 2 alleles, right? Like big A, little a, that kind of thing. So really it's 26 different fragment possibilities. And so then you compare those with different profiles and you can match up who's who. Because 26 is enough to make everybody have a different combination in the world, except for your identical twins. But everybody else has different combinations, which is how you make a genetic profile. Oh, and how does DNA move? DNA moves because DNA is negative, like we talked about, the phosphate's negative. So DNA moves towards a positive. So if I have a picture of a gel or something like that, you're looking, the positive side is where the short stuff will be. The short stuff will be close to the positive. The larger stuff will stay close to the negative. So here's the idea of the different sites. So here's like the sites you would make PCR of. So you can see there's different there are different lengths. So site 1, in this case 1, 2, 3, 4, 5, 6, 7, has 7 of these repeats on both of them. STR site 2 has, I don't know, 7 or something, 8 on the top, but it has something like, I don't know, 15 on the bottom. So that would mean that it has different copy numbers, which means that DNA doesn't match up. And then this is on page 132. is what your gel would look like from that previous picture. So page 132 is what your page 131 would look like. You can see that it has the same size for that orange one, and it's shorter, versus the purple has 2 different sizes.
And so that's how you can tell that they're not the same person?
Yep. This is also how you could tell, like, you do paternity testing like this, or maternity testing, if you didn't know that from the birth, because you're looking at, you'd see like half of the alleles overlapping if it was a parent, because it gets half the sizes from mom, half the sizes from dad. So if it matches with half the sizes, then that's a potential parent or not. All right, I'm just going to write, and I'll do some of the gene expression. Okay. Let me.
Are you on a different page?
I am just going to draw it.
Okay.
Oh, I bet I know what's happening. Oh, this is probably it. Okay, DNA sequence. This is why I was doing weird things. I hit it on the touch, and so every time my hand hit, it was thinking that it was trying to read that at the same time. Okay, ATCGGCA. This is just one tiny part of the DNA sequence. So if we think about our other side, we match up the complementary DNA sequence, that's what the T is for DNA. So A goes with T, T goes with A, C goes with G, G with C, G with C, C with G, and A with T. So that's our double strand DNA. So, what's made from the DNA sequence?
The genes.
Oh, this is true. This is part of a gene. Okay. And if we are going to read that gene to make a protein, and say it's a protein coding gene.
The codons.
We've got to get to the codons. And when we talk about, there are codons in the DNA, but mostly we talk about codons, they're going to be what are in the RNA, because the codons are the groups of 3 that are read by the ribosome, and that's read from the RNA. So in order for us to get to being able to read the protein, or to make the protein with the ribosome, we've got to get to the RNA part here. And so you can actually make all kinds of RNA. There are genes that code for ribosomal RNA, rRNA. There are genes that code for tRNAs. There are genes that code for microRNAs, for small interfering siRNAs, link RNAs, piRNAs. So the genes code for all the types of RNAs. But the only one that's used to make protein is the mRNA, the messenger RNA. If not, then the RNA just exists. So a DNA sequence gene can code for an mRNA, or let's write this here. It can also code for other RNA types, and then those RNA types just go do their job. So there's nothing after this for those. Interestingly, some of those RNA's jobs is to actually regulate the mRNA. So we talked about microRNAs and siRNAs. Their job is to go back and regulate mRNA. So, and ribosomes use the rRNA in order to read the mRNA, and tRNA is matching up with the mRNA. So RNAs are all kind of together. RNAs are very versatile. DNA is pretty boring, honestly. It's a double helix. It has like, it gets modified, but it has relatively low variety in shape. It doesn't do anything except for hold information. RNA, on the other hand, does a lot of stuff. I mean, it can hold information like mRNA. It can do reactions like rRNA. It can be a regulatory like microRNAs or siRNAs. So it's a pretty neat molecule. Okay. So the mRNA, now we have to know which one's the template strand here. So let's say the template strand of this DNA is the top one. Let me draw like this. It doesn't have to be. So for every gene, there's only one strand that's ever read. But for different genes, it can read different strands. So one gene might be on the top strand, the other gene's on the bottom strand. So there's not like one strand of DNA that's template and the other one's not. But for every given gene, there's always one strand that's template and the other one's not. Oh. Let's make this a G. Sorry, it would be better. Yeah, this will be good. Okay, so if this top strand is the template strand and we're building RNA from it, it's going to match up the RNA and the DNA. So RNA is built 5' to 3' because of adding only onto the 3' end. So let's say we're matching up here. This is how I want this to happen. This is not how I want this to happen. I'm sorry. Let's just change these. And we're going to make the bottom strand the template strand. Okay, so if the bottom strand's the template strand, the mRNA is built 5' to 3'. So it's going to start reading, and what it's going to read, so first of all, this process is transcription. And who's going to do it? The RNA builder, which we call RNA polymerase. We also need transcription factors, and they help get the RNA polymerase onto the strand. So transcription factors help start the transcription. And different transcription factors are in different cells. That's why genes are off and on in different cells. So RNA polymerase is everywhere in all the cells. So why aren't all the genes expressed in all the cells all the time? Because only certain transcription factors are in certain cells. And you have to have combos of transcription factors to work. So it's like you have to have 3 transcription factors, let's say, to turn on a gene 1. And only your eye cell has those, but not your liver cell. Well, the genes are present in every single cell of your body. Why is it only on your eye? Because you have those transcription factors in your eye. Why not? And not in your liver. Why? Because they're coded for by genes, and those genes are on, and it just kind of chains back to that. And you're like, well, how does it start? The mom in her egg puts certain transcription factors in certain spots. And so when the cells are dividing and like the cytoplasm divides in half, well, only one of the 2 cells have certain transcription factors, and they're like anchored in regions. And so it ends up different cells in that beginning embryo have actual different transcription factors that start different gene expression, and then it cascades down from there, and so you can get variety. So it's pretty cool. Yay, mom eggs. So that's how that works. Okay, so 5' is going to match up, and so the template strand is on the bottom. So what goes with the A or the T? Got an A, and then we go with the A. Now this is RNA, so what do we put in matching with an A?
A U.
A U. Then we got a C, which is a G. another C, and another C, which is 2 more G's, and then a G, which is a C, and a T, which is an A. And this is just, I mean, this is not long enough to really be a whole strand, but I'm just making it up. Let's add to these RNAs just because I want you to make sure you know this. So our RNA is going to act in the ribosome. I mentioned this already. We've got micro and siRNAs that regulate gene expression. PiRNAs and LINK can do stuff like that too. We've got tRNAs that help with translation. Etc. Okay. So then we've got some mRNAs, though some genes code for mRNAs. So what's going to happen? Then we have, and really this is pre-mRNA. Let's get that correctly in eukaryote. So pre-mRNA meaning RNA before it's processed. In eukaryotes, then we're just going to splice. We're going to slice those introns, or splice the introns out. The introns are the in-between parts, and the exons are the expressed parts. So exons stay in, introns get cut out. And then everything gets, all the exons get put back together, so it's a continuous RNA. Then you get the 2 tails put on, you got the 5' end gets a cap, you got the 3' gets a bunch of A's. Splicing can happen in different combos. So you might have certain other exons cut out, so you end up with different exons in the final product. So that's alternative splicing. So you've got this processing happening here, and that's in eukaryotes only. Then you have translation, right, which is where you have the ribosome jump on the RNA, and it's going to read it in groups of 3s. Those are the codons that you were talking about. So it always jumps on the 5' side with that cap, the ribosome. And it goes and goes along the RNA, because there's some beginning of RNA that has, that's not actually translated. And it goes, though, until it finds the first AUG. So the first AUG is going to be the start codon. And then it reads in groups of 3 from that. So whatever the next group of 3 is. So for like ours, AUG is the first one. And then our next grouping is GGG. And then our next grouping is CA. And then I didn't write one in. So we'll just do, oh, I take that back. AUG, GGC. A, blah, 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 etc. until you get to a stop codon. And there's 3 different stop codons. And then the ribosome, the mRNA, and that all that stuff falls apart, or disassociates. So how does this work? Well, each codon codes for a certain amino acid. So as the ribosome is going along reading, it's matching up the tRNAs and the polypeptides. I'm not going to really draw this too much in here, but you got the tRNAs. This one's leaving in here, and you have this polypeptide that's growing out of the ribosome. And it's from reading those codons. You got your mRNA in here. This is a new amino acid coming in. These are all amino acids. And as it matches up with the codons, it brings in a new amino acid, matches up the codons, and just the tRNA is what matches the codon with the amino acid and brings the new one in. And it adds it to the bot, like the chain gets added to the new one, it shifts, the chain gets added to the new one, it shifts, the chain gets added to the new one, it shifts over and over. And I do have in the all what is it called? The all study material, something like that. So like in all study material, I have all the I have videos linked for all these different topics. So there are videos for transcription and translation and DNA replication. So those might be useful for you to take a look at, the animations, now that you can review those. So each one of these codes for a different amino acid. So like AUG always codes for methionine. So that would be the first one put in. Then the next one, GGC, we'd have to look it up on the codon wheel, but GGC, and then you'd know what amino acid. Then you have A something something, and then something something something, and then you'd have your stop codon. Let's say this would be a tiny polypeptide, too small to fold, but that's the idea here, is that you'd have this polypeptide after you have the translation. Okay. And then the important part here is that you have the certain R groups. So remember, each amino acid has a different R group on it. So let's say some R groups might be methyl groups, and some R groups might be hydroxyl groups, and some of them might have amino groups in them, which have a positive charge. And then remember, the hydroxyl groups are polar, so they have partial positive, partial negative charges. Methyl groups have no charge at all. They're hydrophobic. And we might have, did I say carboxyl? I meant hydroxyl. So now we do carboxyl. Carboxyl groups have a negative charge on them. So we've got all these different R groups, and that's what causes the polypeptide to fold in various ways. So it depends on the order. So the order of the R groups, the sequence of the R groups, is what dictates the polypeptide folding. So let's say we have a polypeptide chain with positive R group here, and then down the chain it has a negative R group here, and then another negative R group. Well, maybe the distance between these two R groups is the right distance, and you're going to have a fold in there. So you'd have like a fold where you'd have that positive R group and negative R group. And so this would be the kind of question I would ask you. What kind of bond do you expect to happen between those two R groups if it's a positive and negative? then we have to go back and think, ooh, what was the positive negative of our group? So that kind of group, that kind of bond here, we would expect an ionic bond. Ionic is between total positives and total negatives. So that would start giving the polypeptide three-dimensional structure. Okay. So what if, though, we had a little bit different sequence? And this could be a mutant sequence, let's say, or just a different gene. Let's say it's a sequence here. Well, now it's a positive and a positive. Those are not going to fold, but we have a negative here. So maybe, and this is like simplified, maybe even like the ionic bond happens there between those guys. And the positive is actually down further because it's repelling that other positive. This is just a made up scenario, but there's our ionic bond. So you see the fold is happening differently. So we end up getting different folding, which we know means different function. And then if you have a polypeptide that's too short, it can't even fold, so you have no function. So that's that with those mutations that cause really short polypeptides, early stop. Those are nonsense mutations. That could lead to a totally non-functional, non-folded polypeptide. If you have substitution mutations, where you're just changing out ATCG, which ultimately changes the codons. Remember, codons might code for the same amino acid, different ones. So that would be silent mutation. Or if it codes for a different amino acid, that would be a missense mutation. If it codes for an early stop, that's the nonsense. And the reason those matter is because of the folding, or it could be in a regulatory region. So if you have a change in the amino acid sequence in this regulatory enhancer region, not amino acid, nucleotide, DNA. The gene is down here. And this turns off or on the gene. Usually we're thinking about it turning on the gene, so it's regulating that gene. This is where the transcription factors bind that I was talking about before. And they could turn on the gene. So if you change the sequence in that regulatory enhancer region, that may cause transcription factors to bind more or less, or change which transcription factors bind. And that can change whether the gene's off or on. It can also change which cells the genes are off or on in. So sometimes there's different enhancers for different tissues. So it can change where it's expressed. It can change like how much it's expressed. It could change whether it's expressed at all, or it could change when it's expressed. The timing might be different. So that's another way that DNA mutations, and actually this is more common than changing proteins, is actually mutation in the regulatory enhancer region will change how much a gene is expressed where or when. And that can change phenotypes often without killing the organism. Unlike a genotype, a change in a protein, if you lose function of a protein, it can be fine, but often it's a problem, whereas changing regulatory regions might not be. So that's another way mutations can happen, not in a gene, but affect the outcome of the phenotype. So you have a cell that has 2 chromosomes in it, let's just say. I mean, there's never any cells with just 2. You can have more than that, but for the sake of ease, let's do 4. I'll do 2. Okay. So during meiosis 1, you have these, first they duplicate and everything.
Mm-hmm.
But when you have them line up, they're lining up with their partners, right? So you have, I'll draw two big ones. We'll make two big ones. One red big one, or pink big one, one purple big one, and then two little ones. So the big ones line up together and the little ones line up together. This is actually, the pairing happens in prophase 1. The lining up happens in metaphase 1. So they're with their homologue, or their partner. And so if we think what happens after that, they're going to separate their partner. This is, I'm not drawing crossing over or anything else, which does happen. So you'd actually have little chunks of purple on the red and the red, or the pink and the pink on the purple. But you end up with this situation here. So if we're going to look at ploidy, we have one big and one little, right? And one big and one little in each. So one of each is 1N, or we just call it N, which is haploid. And this cell's haploid. So you have two haploid cells from a diploid cell. But the problem here is you can't be done because you have double the amount of DNA, because your chromosomes are still duplicated. Okay, so that's why you have to have the second round. So this is meiosis 1. That's meiosis 2. Meiosis 2's job is to separate the sister chromatids. So then you still have haploid cells, one big and one little, but they now have unduplicated chromosomes. And so you can see this is like what we had at the beginning, except for just with one big and one little instead of two bigs and two littles, which is why they're haploid now. So they have half the genes. Does that make sense?
Yes. I was just, well, my brain was thinking that because there was still not exactly a pair, not the homologs, but two in the picture. There's the two chromosomes. I was still thinking that for some reason that's
diploid. Right. That's very common. But it's just haploid and diploid, or triploid, or tetraploid. It's all based off of how many copies of that chromosome type do you have. And, a sister chromatid doesn't count as another copy of the type.
Yeah.
Yeah. Because those should be identical, basically. So, yeah. So that's how that works. It's a good question, though. I mean, most people think it's not haploid until the end, but it is haploid after the first round. Okay. All right. We looked at ionic bonds, but we'll see that proteins can be held together by not just ionic bonds, they can be held together by other bonds too. Okay, so remember covalent bonds, co means together, or shared. So they're going to share electrons in the bond. To fill their valence. So they share electrons. And that's what bonds do. Covalent bonds. Ionic bonds are just between charges. Hydrogen bonds are between partial charges. So they share electrons to fill the valence. So that's what the carbon, oxygen, hydrogen, nitrogen, CHON, you know, those 96% of all living matter is made of those four atoms. That's the kind of bonds they're making, covalent bonds. The next most important ones are sulfur and phosphate, so sponge, some people do, but carbon, hydrogen, oxygen, nitrogen are the 96%. And you see that now that you've looked at the molecules and see how, you know, most of them are made of those four. Okay, so we've got two types of covalent bonds here. We've got polar covalent bonds, which are due to unequal sharing. And why is that? That's because there's a difference in electronegativity, how strong the atoms are. And so one of them hogs, so you got an electron hog, if you want to think of it that way, that keeps the electrons on it more often. Doesn't rip them off, but it keeps the electrons on it more often. Okay? And so this is going to lead to partial charges in your bonds. You're going to have polarity, a partial negative, partial positive. You always have to have both. So one on one atom, one on the other. So let's see where we see that. So we have partial positives, and write this in different color. Okay, we've got partial positives and partial negatives. So which one is going to have the negative and the positive? Well, you know electrons are negatively charged, so that means the partial negative is going to have more of the electrons. So who is highly electronegative and who's an electron hog? Oxygen, when it's bound with hydrogen. So the oxygen is going to be the partially negative, the hydrogen's the partially positive. So that's a really important bond, this OH bond. Another one is the hydrogen-nitrogen bond. Same thing, the nitrogen is the electronegative hog, so it's partially negative, versus the hydrogen, which is partially positive. Those are the two most important polar bonds in biology. So what these guys lead to are dipole-dipole interactions, which are due to the opposite charges. Now, it's not within a bond, but it's between molecules now. Okay, so for example, you have a molecule here, and part of it is a hydroxyl group, so an OH group. So let's draw this like this. The oxygen is the partially negative, and then we have the bond with the hydrogen, which is the partially positive. And then we have another molecule over here. Let's say it has a hydroxyl group as well. We have the partial positive, partial negative on the oxygen, and the partial positive on this hydrogen. So where is this dipole-dipole interaction going to happen between the hydrogen and the oxygen? Now you won't get both, like it's not going to do both because the geometry doesn't allow that. But between two of those, you can have a hydrogen bond. So it's called a hydrogen bond because there's a hydrogen involved in it. That's a type of dipole-dipole interaction, a hydrogen bond. And why does this matter? Well, water, this is what gives water its properties of being sticky, so cohesion, adhesion, of regulating or moderating temperature, making sure it keeps things from getting too hot or too cold because of these hydrogen bonds breaking and making. It's a good solvent because it can get in between other hydrogen bonds. It can get in between ions, like charges, and then it's also, it floats. In its ice, which is the solid form of water, floats in liquid water because the hydrogen bonds they like get stuck and they keep the water further away from each other. So all the water properties are due to hydrogen bonding. DNA strands are held together by hydrogen bonds, the helix. So these hydrogen bonds here are between hydroxyl groups and groups with amino or NHs on them. Also some groups with C double bonded to an O. In this case, we have oxygen again being partial negative and carbon being partial positive. So those kind of groups, because they have partial positives and negatives, hold DNA strands together. The same with RNA three-dimensional structures. So mRNA is just a linear molecule. But other RNAs, like our tRNA, the structure which is critical for, probably not between the loop, critical for its function is held together by dipole-dipole interactions. Other RNAs also have three-dimensional structures. We have protein structure on multiple levels. So you've got the secondary structure, the alpha helices, and beta pleated sheets. Both of those are held together by hydrogen bonds between the backbone. You have the tertiary structure, that's the three-dimensional structure of the alpha helices and beta pleated sheets in some disorganized regions. So that is all held, I mean, not all of it, but some of it's held together by hydrogen bonds. And then we actually have the quaternary structure or quaternary structure, which is where you have your subunits. So maybe you have two types of subunits, so you have 4 of them total, like in hemoglobin. So those parts can be held together by hydrogen bonds too. That's the quaternary structure, which some proteins, many proteins have, but not all of them. So that's another reason that this hydrogen bonding is important. Why else? That we've talked about. Ligand, which is the signaling molecule. And it binds to a receptor, right, in signaling. Many times, that binding of the receptor and the ligand is held together by hydrogen bonds. Let's say this receptor maybe is in a cell membrane. Well, that's not to scale at all, but let's just do it like this. Okay. So that's a plasma membrane and the receptor is embedded in it. It's a protein. And the ligand is held in place in this receptor binding site by hydrogen bonds. And then the same sort of idea is between a substrate and a ligand, not ligand, substrate and enzyme in the active site. So let's say the active site is here. Let's try like this. Here's your substrate that's going to bind in that active site, and it can be held in place by hydrogen bonds. You could also have competitors. So not the substrate, but a competitive inhibitor. In the enzyme active site that could be held in place by hydrogen bonds. Now, if it's held in place by covalent bonds, then it's stuck there. And that would be an inhibitor, that would be like a poison. Most of the time, we should have like these sorts of things being temporary. That's what the hydrogen bond, that's why it's good. Because they stick and then they unstick and stick and unstick and stick and unstick. So not covalent bonds stuck until a reaction happens. Hydrogen bonds are just sticking and unsticking. And that's really important because unsticking stops the signal, right, for a ligand receptor. The substrate needs to unbind after the reaction happens in an enzyme because it needs to go away so the enzyme can continue working. If it's stuck there, then the enzyme can't do its next job. So both of these are temporary. So polar molecules and charged, so polar have partial charges. Molecules can have charged ions. Ions are atoms with a charge. Or molecules that have charges. All those can interact well with water. So that's a consequence of their polarity or charge. Because water is polar, and that's because of the polar bonds. So these unequal sharing of electrons, the partial negatives and the partial positives. So that means anything else that has partial positives, partial negatives, or positives, negatives is going to interact well into it. Is going to stick to water. So that's why you can dissolve polar molecules like glucose in water. That's why cellulose, which is a carbohydrate, all carbohydrates are polar, is good for towels and cotton because it'll absorb the water from your skin because of those hydrogen bonding. That's why you can dissolve sucrose sugar in water. So that's called hydrophilic. That's why you can dissolve salt in water. Salt is a chlorine ion, it's negative, and a sodium ion. So the water can get around those and dissolve it. So that's called hydrophilic. Water loving is what that means. Another place that's hydrophilic here. Oh, and what else does that mean? That means with folding of proteins, okay, you're expecting R groups that are charged to be facing the water. And let's just say this is a protein here. Charges facing the water, you expect partial charges to be facing the water or the outside of the protein. What do you expect on the inside? Hydrophobic groups, which we'll talk about in a moment. Also important here is in our phospholipids, which is the hydrophobic part of the phospholipid, the phosphate group, because it has a negative charge. Hydrophilic, did I say phobic? This is the hydrophilic part. Because the negative charge of the phosphate group allows it to interact well with water, which is why the heads are on the outside. The opposite of that is hydrophilic. This gets to our other. The opposite of that is hydrophobic. Hydrophobic, meaning it does not mix well with water. Now note that this does not repel water. That is a mistake. Not repelling. In fact, it interacts with water. It just doesn't interact well. Water is more stable interacting with other water or other polar. It's not very stable interacting with the hydrophobic. So it gets excluded. So hydrophobic doesn't interact well or strongly with water. Why? Because there's no hydrogen bonds. But it does stick a little. And why are things hydrophobic? The other type of covalent bond, which are nonpolar covalent bonds, and the classic one that we need to know is the carbon-hydrogen bond. Why is this the case? The electronegativity of carbon and hydrogen is nearly equal, so they share electrons relatively equally, which means there's no poles. There's no places where the electrons are more or less concentrated. So that means that there's no charge, no partial or full charges. This is not the same as neutral. Neutral means, in biology, we're usually referring to something at the pH of 7. So I call it no charge instead of neutral. So why does this matter? The most important group that this matters for are lipids. This is their unifying feature, that they have some part of their structure or all of their structure that is hydrophobic, carbons and hydrogens. So for example, like a phospholipid. We just looked at that the part of it was hydrophilic, but there's the part that's hydrophobic, are these carbon-hydrogen tails. Phospholipids have 2 of these tails. I'm just going to draw one in the representation and one in the, I'll draw a bigger head. There we go. Okay. There's that phosphate group's that negative, which is the hydrophilic part. But you can see that this tail here is made, both of those tails are made of carbons and hydrogens, which makes it the hydrophobic part. That means it's amphipathic when it has part hydrophobic, part hydrophilic. Why does this matter? This is why we get membranes the way that they are. Phosphate groups look like this, by the way. It's like attached to the rest of the molecule. At least one negative, if not 2. The other hydrogen might fall off and be 2 negatives, but we've got a negative charge there. That's a negative charge. There's also a glycerol in the phospholipid as well, between the phosphate and the tails, but so it's really like this: tail, tail, phosphate group, glycerol. The most important parts, though, are the phosphate and the tails. So why is this important? This is important for the plasma membrane and membrane-bound organelles. Because it allows regulation of what goes in and out. Without these hydrophobic, hydrophilic parts, that's not going to happen, especially the hydrophobic part. So we've got the phospholipid bilayer. That's part of that's making up the cell or plasma membrane as well as the organelle membranes. So ER, both rough and smooth, Golgi, lysosomes, mitochondria, chloroplast, peroxisome, all those are membrane-bound organelles. The nuclear envelope is also membrane-bound in eukaryotes. All these would be in eukaryotes. Plasma membrane is in prokaryotes as well. So we've got this to where the water is on the outside and the inside of the cell. So the cytosol on the inside, the extracellular fluid on the outside. This is all three-dimensional. I'm just drawing a cut through of it. But it's like actual sphere where the water, the cytosol is the inside of the sphere, and like the water balloon, and then the water on the outside. But we've got these tails in here, and that's the key. The tails create this hydrophobic core. The hydrophobic core is what's selective. They face this direction because the water is on the outside facing the hydrophilic heads, and the tails flip on the inside, and that's the selectivity part. Hydrophobic core. Okay. So what is allowed through directly across via simple diffusion? Simple diffusion means directly across. Versus not. They have to be small molecules. They need to be nonpolar, which means they have no charge, no partial charges. So these are basically going to be gases, small lipids like steroids, which include some hormones like testosterone, estrogen, estradiol, androgens, cortisol. I think cortisol. Don't quote me on that. But some hormones are able to get straight across. Water can get across, but very slowly. Water's polar, but it's really tiny. So it's not great at getting across, but it can. But the most important, the most common ones are these gases. So like, what does this matter? CO2 in and out for our cellular respiration photosynthesis. O2 just goes straight up in and out for cellular respiration photosynthesis. So they just go straight across the membranes, both the mitochondria, chloroplast, and plasma membranes. What does not get across? Anything that's too large needs to be broken down first, or transported by bulk transport. Anything that's polar, so we're thinking like carbs, like monosaccharides, amino acids. These guys are not going to get across. Anything that's charged, so ions, proteins are too big as well. So all that cannot get across directly. So how would you get these kinds of things across if they're small enough? You could use facilitated diffusion with a protein helper to get them across, a channel or a transport protein like a carrier. Both of these, simple diffusion, facilitated diffusion, are going from high concentration to low concentration, down the concentration gradient, so that's how they'd be moving. Just depends if you need a helper or not. If you're doing active transport, it's still the case that it has to be small. You're not going to do active transport and get big stuff across. And in fact, you can't do active transport with stuff that can simple diffuse. You try to actively transport oxygen, it's just going to go back through. So you can only actually do active transport with these things that don't cross themselves. And why would you want to do that? To build up concentration gradients. And we'll talk about that in a moment. Let's just look briefly at an amoeba, so we could see here some transport. So here's this nucleus. It has a little contractile vacuole we'll talk about in a second. And it has solutes in it. So all cells have solutes in them, because they have to have molecules inside and ions and things to do all their processes. So they always have substrates, solutes. So let's say this is an amoeba in fresh water. Okay, so you're going to have water on the outside and water on the inside because the cytosol is mostly made of water. So what happens overall? Well, the water on the outside is fresh water, which means it doesn't have solute in it. So let's think about how we describe our environments. So the environment is going to have a lower concentration of solutes than the inside. So we call that hypotonic, lower concentration. It has to be to something. So to the amoeba cytosol. So the environment is hypotonic to the cytosol, and we can also say that the cytosol, or the cell, is hypertonic, more solute concentration, to the environment, to the fresh water. Hyper means more. Like hyper children have more energy, or hyper anybody. Okay, so what does that mean for water? Well, water is going to, so some solutes can even themselves out, but most of the time, cells are not, like a cell cannot be isotonic with fresh water, because cells have to have stuff inside of them. Isotonic would mean it has only water inside of it. That would never work. So in this case, so what's going to happen? We're going to have water moving towards the place with more solute. So that means more water is moving in than out. So we have a little bit of water moving out, but most of the water is moving in, moving towards the more concentrated solution. Okay, which means what? The cell should, well, it depends. Amoebas don't have a cell wall. That's important. So no cell wall, which means the cell will swell, and eventually it could burst or lyse if we don't have some way to get the water out. And that's why the contractile vacuole is there. It fills up with water and then squirts it out. Which is what prevents the amoeba from exploding. And many contractile vacuoles form. They form, collect the water, fuse with the plasma membrane, squirt it out. Morels form, they collect the water, and that kind of thing. It's the basic idea. So instead of bursting or lysing, they have this ability, freshwater amoebas. But if you put a saltwater amoeba in freshwater, or it might not have this ability, and so it might actually explode. Or even if you put it in an environment it's not adapted for, that could happen. What happens to a cell without a cell wall in an isotonic environment? So you have an environment that's isotonic to the cell. That means water is going in and out equally as much. So dynamic equilibrium there. That means that it's going in with a cell without a cell wall. That's a positive condition, so it's going to stay the same size. It's going to be, I wouldn't say happy, but it's going to be stable. If you have a cell without a cell wall in an environment with more solute, oh, and this environment would not have no solute, the isotonic. Okay, so the environment is hypertonic to the cytosol, or the cell. Or we could say the cytosol is hypotonic to the environment. So where is it going to move? The water moves towards the more concentrated environment, which means it's going to, some of it will move in, but more of it will move out. The net will move out. So this in a cell without cell walls means that the cell will shrivel and eventually die. What about like a plant cell, a cell with a cell wall? So the most important part of the plant cell for water balance is the central vacuole, which can take up a large portion of the cell. So if it's really plumped and swollen, because most of the water's going in, so we have the environment is hypotonic to the cell. That means water overall is going to be moving in, which means that the cell is turgid or swollen, but because it has a cell wall, it doesn't let it explode, which is a positive state, so it keeps it nice and plump. So hypotonic environment with a cell wall is a good state. Stable state. What about a cell with a cell wall in an isotonic environment? Well, the water is going to move in and out at an equal rate. And actually this leads to the cell being flaccid because it's not plumped up. It's not swollen to keep its rigid structure. So the cell is flaccid. You might get your plant wilting, becomes wilted, and this is not the most awful condition, but it's not a great condition. So we'll put a face like this. It's not good though. Hypotonic is better for cells with cell walls. What about in a hypertonic environment? Actually, the plasma membrane pulls away from the cell wall actually. Why? Because in a hypertonic environment, more water is going out than coming in, which means it's losing the water, which means it undergoes plasmolysis, or it plasmolyzes, and then it dies. So the cell wall pulls away, the plasma membrane pulls away, the central vacuole is really small, and it wilts and then dies. So plasmolysis is like after it becomes flaccid and then death. So that's not good. All right, so those are tonicity ideas here about hypertonic, isotonic. Remember, you always are comparing two solutions. You can't be hypertonic on your own. So membrane selectivity allows, why is this important? That selectivity allows regulation of what goes in and out, but it also allows building up of concentration gradients. This is not, maybe I wouldn't say selectivity. Membrane, well, yes. So the fact that it doesn't just let everything go across is what I'm talking about. Why is this important? Okay, what is a concentration gradient? It means there's an unequal concentration across the membrane, more inside or outside. Why is this important? Because it's potential energy. Potential energy is stored energy. And that stored energy can be used to do work. For example, make ATP. For example, fire a neuron. Making ATP both in cellular respiration and photosynthesis uses these concentration gradients. Specifically, the concentration gradient used for making ATP is with the hydrogen ions, H+, or we can call those protons, across membranes. And that gradient is created by the electron transport chain in both mitochondria and in chloroplasts. Okay. So when is ATP made in photosynthesis? The light reactions. Remember, only enough ATP to actually run photosynthesis, not enough to run the cell. Okay, so let's think about where this is happening in the cell. Well, it's going to happen in the mitochondria. Excuse me. It's going to happen in the chloroplast. Okay, so photosynthesis happens in the chloroplast. And this is where we're having it occur. It's in the thylakoid membranes, the stroma. There's all this stuff out here. There's a lot more thylakoid membranes. I'm just drawing like one little stack of them. And it's a double membraned organelle with more membranes inside. So I'll draw it like that. Okay, so in this thylakoid membrane, we have photosystems. We have an electron transport chain, another photosystem, another little chain, and then some ATP synthases. I'm just drawing them all in a row. The chains and the photosystems are in a row. The ATP synthases can be other places, but I'm just going to do it like this. This is the stroma out here, and this is the thylakoid space. Call it that. In here. Okay. Photosystem 2, photosystem 1. This is the part that has all the pigments in it. Captures the light. So light is going to come into both photosystems. And that energy gets captured and it bounces electrons out of their orbital, bounces electrons around until they're captured and basically passed to the electron transport chain. This passing is going to release energy. That energy is now used to pump hydrogen ions across the membrane, or protons across the membrane. That's why you build up the protons inside the thylakoid space. Draw this the other way. Okay, then the electrons are passed to photosystem 1, where they get bumped up again. Electrons are passed to the little electron transport chain, and then they're passed out of the system. So let's look at what's involved here. We have molecules coming in to drop off more electrons, right? Because if these are electrons, they're getting passed around. How are you going to refill the electrons at the beginning of the whole thing? You need an electron donor. That's what your water is. Water comes in with electrons. It drops the electrons off by getting broken. So you've got to break the bonds to release electrons. It's a very common theme. So you break the bonds of hydrogen to release electrons. Those electrons get put into the chain, and then you have you have the oxygen left, and the oxygen binds another oxygen. After the next water is broken, it comes out as O2. So this diffuses out of the chloroplast. can be used in cellular respiration or just released to the environment and then used in cellular respiration for another organism. Okay, so that's the water-oxygen business in photosynthesis. What is going to take the electrons away at the end? We have a molecule called NAD. This is in photosynthesis, so P plus. It's going to pick up the electrons and leave as NADPH. That's going to go to the Calvin cycle. All right, you have all these hydrogens on the inside creating this concentration gradient where the hydrogen's going to want to go to the place where there's less of them. So that's out of the thylakoid space into the stroma. The only place they can exit is through this ATP synthase. It's an enzyme. So as the hydrogen ions exit, the ATP synthase spins, and that spinning action is going to allow it to take ADP, add a phosphate group to it, and make ATP, which goes to the Calvin cycle. Great. And then we should label, this is ATP synthase, ATP making enzyme. That's what it is. Okay. So that's basically the light reactions and how the whole ATP is made there using concentration gradients. And it's advantageous because you see the concentration gradient allows you to make ATP without having to use ATP. Sometimes concentration gradients are, or sometimes reactions are, or active processes are done. Oh, let's label this. This is active transport here. And it's done without ATP, like I mentioned. Some active transport is done using ATP, like sodium-potassium pump. But this is a form of active transport that does not use that. It uses the electrons falling. So that's the process in photosynthesis. Similar process happens in cellular respiration. You can, of course, pause it there if you need to look at that. There's the mitochondrion. Also double membrane organelle. Its hydrogen ions are going to concentrate in this intermembrane space. The other area here in the inside is the matrix. And then some processes happen out in the cytosol, like glycolysis. So where's this gradient working in cellular respiration? It's working in oxidative phosphorylation, the last step. Remember, oxidative means oxygen dependent. Phosphorylation means putting a phosphate group on ADP, so making ATP. So it's oxygen dependent ATP making. Actually, the light reactions, this whole process here is called photophosphorylation, light dependent ATP making. Oxidative phosphorylation, oxygen dependent ATP making. Okay, so very similar deal here. But we don't have photosystems because there's no light involved. I'm just going to draw an electron transport chain and then the ATP synthase. I'll draw the matrix up here. And we'll draw the intermembrane space down here. Okay, so where do we get the electrons from in this case? Well, they're not from water this time. Water's going to be at the opposite end, actually. But this time they're from NADH. Let's see where we get the NADH from. We have out in the cytosol, glycolysis, that gives us a little NADH. Then we have pyruvate processing. We could call it oxidation of pyruvate, which means stripping pyruvate of electrons. That gives us some NADH. Then we have the citric acid cycle, which gives us some NADH. So all of those are stripping electrons from glucose, from pyruvate, from the molecule cycling the citric acid cycle. That gives us the NADH, which drops those electrons off and leaves as NAD plus. Those electrons are going to fall down the electron transport chain. They get passed. Their energy gets released, and we pump the hydrogen ions. This time we're pumping them into the intermembrane space. Should draw another. Building up that concentration gradient, where we have lots of hydrogen ions in the intermembrane space, or lots of protons, not as many in the matrix. Take this backwards again. I guess I usually draw it the other direction of movement, but it doesn't matter. I draw it like this because the bulb part, that kind of lollipop head part, is which spins, is the part that's on the opposite side of where the hydrogen ions go through. So then what happens? Okay, where do the electrons go at the end of the chain? They've got to get taken away. This is the opposite of photosynthesis. So who dropped them off? Water. And what came out? Oxygen. Who's going to pick them up? Oxygen. And what's it going to make? Water. Opposite. This is part of that cycling. So electrons go into the oxygen, and then they end up as part of water. So it's the oxygen plus 2 hydrogens plus 2 electrons. The hydrogens are going to flow through the ATP synthase just like before. ADP and inorganic phosphates are getting put together to make ATP. And in this case, the ATP goes to run the cell. So like all the cell processes that need ATP, that's where this comes from. Let's see. You could call it cellular work. Let's go back up. What happens in the the Calvin cycle? You're going to use the NADPH for the electrons and use the ATP for the energy. And using the energy, you're going to embed the electrons in a carbon compound. That's the point of this whole process. Carbon compound means organic molecule. That was the point. So embed those electrons in the carbon compound, yay. And then that carbon compound is what comes in here in glycolysis. So what do we have here? Carbon compound coming into glycolysis, or actually any of these steps. If it's glucose, it'll go into glycolysis. If it's something, if it's like amino acids, it'll filter into other parts. If it's lipids, they'll get broken down and filter into other parts. So all of those carbon compounds are filtering in there to get the electron strip. So that's what the citric acid cycle and glycolysis and oxidation of pyruvate. They're stripping or extracting electrons from the carbon compounds. So it's the opposite of photosynthesis, where we embed them and we strip them out, then we embed them and we strip them out. Good. So that's basically cellular respiration photosynthesis. Interestingly, so going to an idea about pH now. So in the part of the organelle with lots of hydrogen ions in it, so whether this is the intermembrane space or inside the thylakoid, either one. Let's think about the pH there. Let's go back to that idea. There's lots of hydrogen ions or protons. So what are hydrogen ions involved with pH? Hydrogen ions make solutions more acidic. So the inside of the thylakoid discs is going to be highly acidic, or more acidic compared to the stroma. The inside of the intermembrane space is going to be more acidic than the matrix. And why is that? Because the hydrogen ions are being concentrated there. So remember, and in fact, this is going to be acidic, so it's an acidic pH. So let's just remember acidic, neutral, and basic. Acidic means the pH is greater than, less than 7. Neutral, the pH is equal to 7. Basic, the pH is greater than 7. In acidic pHs, your hydrogen ion concentration is greater than your hydroxide ion concentration. In neutral pHs, they're equal. In basic pHs, your hydrogen ion concentration is lower than your hydroxide ion concentration. Remember, that's actually, these words are different than the words acid and base. These are substances, not adjectives. Acidic, basic, neutral describe solutions. Acid and base are substances, molecules. An acid makes a solution more acidic. Not necessarily acidic, but more acidic than it was. A base makes the solution more basic. So an acid is going to lower the pH, that makes it more acidic. A base is going to raise the pH, that's meaning it's getting more basic. So if you had the pH, a solution with a pH of 4, and then it turned into a pH of 6, what did you add to it? Well, you are increasing the pH, making it more basic, so you must have added a base to it, which made it more basic. Interestingly, look at this though, the pH is still acidic. So it starts acidic, it ends acidic, but you added a base. So a base doesn't mean it gets basic, it just makes it more basic than it was. Just to note that each pH jump is 10 times more or less hydrogen ion concentration. Each pH number is 10 times more. So if it's like 10 molar, it'll be 100 molar. Kind of thing. Let's think about buffers, really important for biological systems because they help maintain homeostasis. Homeostasis is when your conditions are in a steady state, so they're not wildly fluctuating. There's a certain range of pH that is acceptable for different organisms, have different ranges, but it must stay within that range, and buffers allow it to maintain. Homeostasis, steady state. So what does a buffer do? If the pH is too high, it's going to means, if it's too high, which means it has too high pH equals too low hydrogen ion concentration, right? Because high pHs are basic. So what does the buffer do? The pH, the hydrogen ion concentration is too low, so we need to increase that. And what is it? How is it going to increase that? It's going to be an acid. So the buffer acts as an acid to increase the hydrogen ion concentration and lower the pH. If it's too low of a pH, that means there's too high hydrogen ions. What's the buffer going to do? The buffer is going to lower the hydrogen ion concentration by being a base. So the buffer can both be it can be a base or it can be an acid. And that's usually because it goes back and forth between being an ion and not an ion. So like the most famous buffering system in biological systems is the bicarbonate, H2CO3. And it can go back and forth between that and HCO3 minus plus a hydrogen. So it can lose the hydrogen. See, it has two hydrogens here, and then it only has one hydrogen, and then the other one fell off. So it can have them separated or together, separated or together, separated or together. So if you need more hydrogen ions, it's going to separate. If you need less hydrogen ions, it's going to bind them up. So when it's moving in the when it's dropping off its H's, releasing the H's, going to the right, it's acting as an acid. When it's picking up the H's and going to the left, it's acting as a base. And so basically it can go between those two in order to change the overall hydrogen ion concentration of the solution. pH matters because it can get in between. So if we have a protein that has some weak bonds in it that are from charges, so let's say the negative charge and a positive charge, partial negative, partial positive, any of those kinds of things. If the pH is too high or too low, the hydroxyl or the hydrogen ions, the H pluses or the OH hydroxide ions can get in between here and break the bonds. And basically this can make your protein unravel or denature. So that's a denatured protein, an unfolded protein, which means it probably loses its function. So that's why it's one of the reasons it's important to keep pH in a narrow range, so you don't have all your proteins unraveling and then having a function. Yeah, so look over your enzyme stuff. That's pretty straightforward. Energy transformations, we talked about some of them here. Genetics problems, I have a lot of those I've worked online, so I'm not going to do any more of those on this recording. You can definitely go look at me working a bunch of those problems. I have example problems in here as well, so if you go to the slides with those, let's see. I don't know exactly where they are, but you can find them in the PowerPoint. You can actually download this too by pushing this download button. It'll download the PDF. I think this is also in the review information. Then there's the stuff about evolution at the end. I think I've gone over, isotopes, ions, which we've talked about. Viruses, most of you guys did very well in that, so I'm not going to go over that in detail or anything. But you want to know the difference between what viruses have and how they work versus prokaryotic cells versus eukaryotic cells, and then specifically animal cells and plant cells. So differentiate those. Functions of organelles, it'll be like I did on the exam where it's sort of like a problem with the cell, what organelle is messed up, or this cell does this process, what organelle will have a lot of, something along those lines. I do have genetics problems on there. As I mentioned, mitosis, meiosis, people do generally pretty well on those topics. So I'm not going to go over those in detail either. Cell cycle. A little bit about cancer, proto-oncogenes, tumor suppressor genes, oncogenes, metastasis, benign, metastatic tumors, that sort of thing. How enzymes work, how ATP works. So that's what I can think of. But we've gone over a lot here, and you can see how we're like connecting ideas between the chapters. So there will be a decent amount of that on the final, where I'll ask you about something in the context of something else. But just you know the information. So just take it a step at a time, and you'll do great. So I hope that helps.